Good evening and a hearty welcome to all. This is Simran Srivastava from 2nd UG MCZ, your moderator for this session, and it is my honor to introduce our esteemed speaker for today's lecture on the topic Thinking Outside the Cell, Fighting Pancreatic Cancer by Targeting Extracellular Enzyme MMP7. An alumnus of St. Joseph's College, Dr. Vishnu graduated in BSc Microbiology, Chemistry and Zoology in 2009 and then went on to pursue a master's in molecular biology and human genetics from Manipal University and passed with high honor with a 3.91 GPA. He was a student research trainee from 2012 to 2013 at the Tel Aviv Sourasky Medical Center and Tel Aviv University, Israel. Sir did his PhD at the Feinberg Graduate School, Weizmann Institute of Science, Israel, under advisor Professor Irit Sagi. His doctoral dissertation was focused on revealing the versatile roles of matrix metalloproteinase 7 in pancreatic cancer, and he finished his postdoctoral bridging training at the same institute. He is now doing his postdoctoral training under advisor Professor Tamar Geiger, where his main focus is investigating dynamic cell-to-cell interactions over time in cancer using mass spectrometry-based proteomics. His impressive CV boasts several accolades, including Dean of Faculty Fellowship for Postdoctoral Research 2021. Dr. Mohan has 9 brilliant publications to his name, the most recent being based on selective inhibition of catalytic MMP7 in PanNet, reveals secret protein kinase VLK as a novel MMP7 substrate. Not only that, he also has a patent to his name. Dr. Vishnu Mohan is the co-inventor in anti-matrix metalloproteinase 7 inhibitory antibody and uses thereof, filed on 10th February 2020. A testament to his training and education, Sir is an incredibly skilled researcher, well equipped in several nuanced techniques like multiphoton microscopy, scanning electron microscopy, antibody purification, and characterization. Sir has also given several presentations in conferences and seminars, including very recently at the 7th FEBS Advanced Lecture Course in Matrix Pathobiology, Signaling and Molecular Targeting, May 2019, at Porto Helai, Greece, titled Deciphering the Complex Role of MMP7 in Pancreatic Cancer. On the non-academic front, Sir has been the residence advisor for international students at the Weizmann Institute of Science since 2014. He has also been a member of the Weizmann debate team, president of the Student Council at School of Life Sciences, Manipal University. He is also proficient in English, Hindi, Malayalam, and additionally speaks Hebrew and German. We are honored to have you here with us, sir. Before the session begins, I request everyone to please refrain from switching on your audios and videos. Any queries can be posted on the chat box and will be asked and answered at the end of the lecture. Without any further ado, I hand over the floor to Dr. Vishnu Mohan.
Hi, thank you for that generous introduction. I'm really privileged to be here. And at the onset, I would like to thank the microbiology department at St. Joseph's College and the whole Kaleidoscope 4.0 team for inviting me to give this lecture. I shall now share my screen. So, are you able to see the presentation?
Yes, Vishnu.
The talk today is going to be about pancreatic cancer, but we are going to approach this really difficult to tackle cancer from a novel perspective which has not been explored much in depth so far. So before I start, I just want to say that I work at the Weizmann Institute of Science here. It's located south of Tel Aviv in Israel, in a city called Rehovot. And the Weizmann Institute of Science is known world over for good research output. It was ranked the 8th recently in the Leiden rankings for quality of research output. It's an institution that has only masters and above, so masters, PhDs, and postdocs, no bachelor degrees. And we'll talk about the institute in detail at the end of my presentation. The talk I'm going to give today is about the work I did for my PhD in the lab of Professor Irit Sagi. Professor Sagi is now the Vice President of Technology Transfer at the Institute, and she heads the Technology Transfer team. The Institute is really special in the case, in the arena of tech transfer. So conjoined to the large campus of the Institute is a dedicated science park, which has many established companies and companies who are in incubators under venture capital firms. So the Weizmann has a good mechanism of churning out research all the way from the bench to the bedside. So the introduction starts with what is pancreas, where is pancreas in our body, and why are we concerned about pancreas in the preview of this talk. So pancreas is an abdominal organ, as you all know. It's placed close to the intestine. The pancreas is a very complex organ. It has both the exocrine side as well as the endocrine side. The exocrine part of the pancreas secretes enzymes for digesting food into the small intestine. These enzymes are drained to the small intestine using a duct. And of course, other than the exocrine part of the pancreas, this complex organ has embedded within the exocrine part, the endocrine part of the organ. And the endocrine tissue is together in the islets of Langerhans. And these islets of Langerhans are dispersed within the tissue, and they are really well supplied by blood vasculature. And of course, the islets of Langerhans has alpha cells, beta cells, delta cells, and the beta cells, of course, produce insulin. And these are the organ as such, pancreas, is really central to our daily well-being. And it comes out of a developmental axis together with the liver in a very special manner compared to many other tissues. And this is relevant for us as we go forward in the presentation. So once again, pancreas hidden behind the stomach, if you look frontally, draining its content into the intestine and dispersed with beta cells of the islets of Langerhans. So there are the whole topic today is about cancer. I'm going to talk about cancer. This is what I do, cancer biology, and cancers of pancreas are of various types, depending on the tissue of origin. So we have the pancreatic ductal adenocarcinoma, which are the most common type of pancreatic cancers. Pancreatic ductal adenocarcinoma are really lethal cancers because they develop over many years, but they develop really quietly. They don't have any outward appearances, not even indigestion or pain, till you reach stage 3 or stage 4. In stage 3 and stage 4, the tumors would have already metastasized to its locations where it has tropic attraction to. As a result, after average diagnosis, the survival is really low. It's between 1 to 2 or 1 to 3 years. So pancreatic ductal adenocarcinoma is a solid tumor disease that we haven't gotten a full hold of. Many decades of research has not produced, therapeutic substances or methods that can really be helpful in extending lifespan after average diagnosis. So it continues to be a major issue to tackle in cancer. And along with ductal adenocarcinomas, a smaller fraction of tumors that arise in the pancreas come from the cells of the islets of Langerhans. These can be either functional or non-functional. So let me take a moment to explain to you what that means. Functional tumors of beta cells are called insulinomas. They are called insulinomas because when the cell number of beta cells proliferate and increase, they produce a lot more insulin. So you immediately start to see effects in the body. So you would probably go to the doctor. The patient would probably go to the doctor complaining about lack of sleep, a lack of rest, problems with all the problems that are associated with blood sugar control. And then the doctor would realize there is hyperinsulinemia, and then would ask you to go for a scan to see what's going on, and then that's how it is detected. There are fundamental differences between these two cancer types. Ductal adenocarcinomas, as I told you, progress really silently, but eventually they show up in a very late stage and very difficult to treat. Most of the functional endocrine tumors, on the other hand, let it be glucagonoma or insulinoma or any other tumors of the cells of the islets of Langerhans, they come out pretty much early in routine checkups, and they have slightly better overall survival. But I only say slightly better because it's in years, not in decades. So on the whole, pancreas as an organ is a very difficult organ to tackle when it's affected with cancer. Just to say a few names, we lost Steve Jobs to beta cell cancer. Aretha Franklin, famous singer from the US, was also lost to pancreatic cancer. And recently, our ex-defense minister, Manohar Parrikar, we also lost him to pancreatic cancer. So it's a disease that is only increasing in its footprint, and we need to tackle it with methods that are different from what we have been doing so far, so we can be more successful. That brings us to the focus of Irit Sagi lab, the lab that I did my PhD in. So Irit's lab is really concentrated on what we call the frame of the picture, because we basically look at the extracellular matrix. So all the material that is outside the cell. The matrix is a really complex system. It's actually a very dynamic system. It was thought for many decades to be a really, just a cement that holds all the cells together. But it really is not true. We have increasing evidence that the matrix is a dynamic reservoir, and it has many functions when it comes to both homeostasis as well as disease. You can see here in this review that I published, there's different functions of matrix associated with a cancer cell. There's cross-linking that helps them to migrate. There's altered cell signaling cues. There's hosting of the cancer microbiome. And there's, of course, also neoangiogenesis that is important for cancer to expand. Most of the ECM of collagen. Collagen actually is the most abundant protein in our body. It is there in all these tissues that I've shown here, mentioned here. And collagen, along with fibronectin and laminin and numerous other proteins, make this really complex extracellular matrix network. The network can be structural, or it can be basement membrane, and it can host a large array of cells when it comes to tumor. This is actually a picture that I took in the scanning electron microscope. You can see what we believe from the morphology is a fibroblast cell holding going on a river crossing or something. It's holding on to the collagen helixes, and this is within the capsule of a pancreatic neuroendocrine tumor. So showing again that for recruitment of cancer-associated cells, such as fibroblast, the ECM plays a very central role. So to recap, the ECM provides anchorage to the cancer cell. It's a migration barrier in normal circumstances, but it provides migration tracks for cancer cells, helping them to move out of their primary locations and do intra and extravasation, resulting in metastasis. It's also a signal reservoir. It's a co-receptor, low affinity co-receptor. It also presents other cells. For example, if your immune system is trying to attack the cancer, it provides proximity between the T cell and the cancer cell. And of course, it produces a lot of functional fragments that are important for signaling or other activities. What is really important for us to remember is that as we age, as human beings and as mammals, as animals, we generate a lot of occult tumorigenic sites. Many of them are attacked and removed by the immune system. It's only the rarest of the rare that escapes this immune response, and some of us get cancer. So while appreciating the robustness of the immune system and our surveillance against cancer cells, we also want to appreciate that those cells that have escaped the surveillance, how complex they are and how manipulative they can be towards the end of tumor growth. So I want to try and talk today not in a lot of technical detail, so that in the wide range of audiences that are here, it's more comprehensible. So when I analyze tissues, we can analyze tissues for extracellular matrix imaging using a particular type of microscopy. It's called two-photon microscopy. Two-photon microscopy has a mode in it in which you can use imaging of these, use it to see the images of the ECM. This mode is called second harmonics generation. Second harmonics generation imaging provides ECM without any staining. So you can take naive tissue. In this case, what you're looking at is human pancreatic ductal adenocarcinoma sections that we acquired from our collaborators in Sloan Kettering Cancer Center in New York. And these fresh frozen sections were exposed to two-photon microscope to generate images of the ECM. So what you see in green and in red here are two different ways of looking at the ECM, in the reflected and the transmitted signal. And you see the combined signal here. And what you'll really appreciate is that when you move from the adjacent normal tissue to the tumor periphery to the tumor core, in ductal adenocarcinomas, it's completely enriched with ECM, increasingly. And you see that the orientation becomes really straight. And this is indicative of how good the ECM has reprogrammed, allowing cancer cells to move along it like a highway, like a tract. So you see here fiber orientation getting really concentrated into one peak. And the beauty of this imaging is that we don't do any other staining step. It's completely naive tissues, which is imaged using the two-photon microscope. But the underlying factor that we can understand from here is that ECM is dense in ductal adenocarcinomas. And often clinicians, when they take out the tissue, they'll tell you that ductal adenocarcinomas look like a white mass, whereas neuroendocrine tumors, they look like a red mass because they have a lot of leaked blood vasculature and relatively lesser amount of ECM. This is another image, comparative image, which is created in scanning electron microscopy. You can see here, when you go from adjacent normal to tumor periphery to tumor core, we can see in detail the collagen scaffold, how it is coming undone, and it becomes a chaotic mess of fibers in the tumor core. And this is actually direct evidence for ECM remodeling that is happening on the behest of tumor cells. So extensive ECM remodeling is only possible because there are enzymes that help to chop and rejoin these fibers. And these enzymes are called as matrix metalloproteases. There are many enzymes that work on ECM proteins, but matrix metalloproteases and crosslinkers called lysyl oxidases or LOXes are the most important enzymes in the process. If you go from adjacent normal to tumor periphery to tumor core or tumor center, you see that lysyl oxidases are cross-linking enzymes, the staplers of ECM, as we call them. They get really localized to the fibrous tract. And we know from previous imaging that these tracts are basically rich with ECM and fibroblasts. And when you look at the tumor cells, they get enriched with matrix metalloprotease enzymes. And in particular, shown here is matrix metalloprotease 14, or what we call membrane type matrix metalloprotease. Another enzyme which is of real interest for us in this active ECM remodeling process is MMP7. MMP7 or matrix metalloprotease 7, when you look at stainings using commercial antibodies, is really enriched in all these sections of tumors. These are separate patient tumors, patient 1, 2, 3. And you see that there's very high expression of MMP7 in these tumors. So I want to gather your attention to MMP7. MMP7 is a power-packed mini MMP. And this MMP is a mini MMP because unlike other MMPs, it lacks a hemopexin domain, which is indicated here in orange. It's an MMP that has the catalytic site. The catalytic site, of course, has a zinc metal. The zinc metal catalytic site is a characteristic feature of the 24 or 25 MMPs that are present in mammals. And along with that, it has a pro domain. And most of the MMPs are created with a pro domain, and the pro domain has to be shed at the site of activity for them to act as a chopper or a shed-ase or remodeler. So this is interesting because most MMPs have this really tight regulation, not only in terms of expression, but also in terms of localization as well as activation through shedding of the prodomain. And the reason for that is these are really potent enzymes. So if you put MMPs with collagen that are active, they will start chopping and doing their job right away. And we don't want, in our tightly regulated homeostatic system, we don't want these enzymes to go berserk. But the cancer basically uses this enzyme to its own end, like it uses many other mechanisms. And I just want to gather your attention to the substrates of MMP7 as an enzyme. It has substrates from the matrisome. Matrisome is a word we use for proteome of the matrix. It's all the components and interactors of the extracellular matrix. So here you have proteoglycans, laminin, fibronectin, and other MMPs also get cleaved from the prodomain and activated by MMP7. But MMP7 also has a lot of signaling substrates. And this is really interesting because signaling substrates means you have amplification effect of MMP7 levels and activity. And many downstream processes get affected by MMP7 activity. So these are the substrates that are relevant for us in cancer as we speak and go forward. So if you look at the median expression of tumor and normal samples for MMP7, you appreciate that from the TCGA data there is very high expression of tumors, like MMP7 in tumors from pancreas. You see here pancreas in highlighted. But when you compare to the normal pancreas, the expression is comparatively really low. And for many years, there's been talk about using MMP7 as a particularly good marker for indication of survival. So especially recurrence-free survival. So MMP7 seems to be an important player. And that's when we jumped in to use the expertise of our lab. So Iritsagi lab works on creating inhibitors against these potent enzymes. So these inhibitors can be of different types. There are small molecule inhibitors. There are, small molecule inhibitors are chemical agents, small in size, as the name indicates. You have active site antibodies. So we already come to the realm of biologics. So producing antibodies against the active site of the enzyme. But this has a negative connotation to it. Active sites of most MMPs are highly conserved, not only in humans, but across the species. Raising antibodies against active site means you may not have specificity to block one MMP. You might be blocking multiple MMPs. Then you also have mimicry of naturally occurring inhibitors of MMPs called TIMs, tissue inhibitors of metalloproteases. So they are basically engineered endogenous inhibitors that can be produced, and there are these inhibitors produced in the lab as well. And finally, the new generation of allosteric antibodies. These are antibodies that bind to a site far away from the active site, but because of the binding and the changes it creates in the structure of the protease as a whole, it is unable to do its binding activity on substrates. So the antibody that I'll be talking to you today is somewhere in between an active site antibody and an allosteric antibody. It is not an endogenous inhibitor, engineered endogenous inhibitor, but it's a version better than the active site antibody, but it's not yet a full allosteric antibody. So our aim was to generate an anti-MMP7 antibody with high affinity and selectivity, and then to validate the impact that MMP7 inhibition will have in PDAC and PANNET cells, because we know that MMP7 is high and playing a role in ductal adenocarcinomas and neuroendocrine tumors of the pancreas. And we wanted to utilize this antibody as a tool to identify new substrates of MMP7 enzyme and to validate it, because this is a holy grail in the field, to come up with new substrates for existing enzymes to understand the biology better so we can target them or use them better. So I just want to walk you through generation of the antibody process. Here you can see that the immunization was done with a mimicry molecule called zinc tripod. It mimics the active site of this enzyme and also the active enzyme in an alternative way. So we had zinc tripod 3 times, enzyme later. Basically, we come up at the end with candidates for fusion, and we fuse these cells to create hybridomas. And from the hybridoma culture, we isolate antibodies in large-scale bioreactors. So you'll appreciate that the ELISA from the bleed of the mice has dual specificity. That's because it's a concoction of antibodies, and then we start to expand particular hybridomas with monoclonal nature. So you see here that conformation specific antibody against anti-MP7 this is the paper that we published recently about this antibody. It's out there for all of you to access. I think it's an open access journal. And I will be explaining a little bit of the results that we described in this paper today. So we see that the ELISA to determine activity was really good in terms of EC50. We got in nanomolar range. when we try to do a fluorogenic peptide assay. So I just want to explain briefly what this assay is. You have in a plate enzyme along with substrate. What sort of a substrate is it? It's a fluorogenic substrate. It's a synthetic peptide with a fluorophore at the end and a quencher at the other end. So if the enzyme is active, it makes a cleavage on this peptide, and then the quencher is released. That means there'll be a signal. So high signal means high activity of the enzyme. And in the presence of the antibody, we selectively inhibited MMP7 from the family of other MMPs with relatively similar active site, giving us a lot of confidence that it's an enzyme targeting antibody that is very much against MMP7 and not against other MMPs. So this is a, if I can change that. Yeah, you see here a video of the modeling that we did. We were not able to co-crystallize the antibody and the target enzyme, but nevertheless, there was existing structures of the enzyme. So we crystallized the antibody and we did modeling analysis. And when we do the modeling, we saw that the least energy interactions are at sites very close to the active site, but not at the active site. So it's not an active site binding antibody, but it's binding close to the active site. And it's basically creating steric hindrance for other substrates to enter. So in green is the antibody and in gray is the enzyme. So when you come to the biology side of things, and when we started treatment, we realized that there is an immediate effect in survival of the cells. Also, without using the antibody, when you do silencing, lentiviral silencing, we see that there is a drop in the sub-G1 peak when it comes to MP7 silence cells, showing that there's, there's an increase in the sub-G1 peak, showing that there is increased apoptosis. That means cell survival is being affected. There's increased apoptosis in the absence of MP7. So we confirmed this using an Annexin-5 FITC assay. It's an assay that is routinely used using FACS for characterizing cells that are undergoing apoptosis. And you basically have to count Q1 plus Q2 in each of these graphs, and then you get a comparative level of Annexin-5 that is there, and it indicates increased apoptosis. So with increasing concentration of the antibody, we saw there was increasing apoptosis that was happening. And this was confirmed using Western blot against Fas ligand. And the reason we chose to look at Fas ligand is that it's known that Fas ligand and receptor are substrates of MP7. And we realized that this could be the pathway in which it's stabilizing. So basically, if MMP7 is active, it cleaves the Fas ligand and does not allow the cells to induce cell death on each other. But when we stop the MMP7 with the antibody, then the ligand receptor interaction between cells are active and they spontaneously start apoptosis. Alternatively, we know that E-cadherin is also a good substrate of MP7. And when we prevented MP7 activity using the antibody that we developed called GSM192, the antibody prevented closing of the wound in a wound assay, as well as migration across the Boyden chamber. But the most important role that MMP7 plays in pancreatic cancer is in chemoresistance. There's a lot of evidence about how pancreatic cancer, through the Fas receptor ligand system shedding or through other methods, are helping the cancer cells withstand chemotherapy and cytotoxicity from chemotherapy. So we checked in the presence of our antibody how the chemotherapeutics work. And we saw that if you add the antibody, the IC50 compared to vehicle control comes drastically down, significantly down. And this is true for different cell lines. As you see here, SPC1, BXPC3, these are two different cell lines from human PDAC. And it's also true for two different agents, oxaliplatin and gemcitabine. For us, we really wanted to sum it up in a paper, and that's the paper that I showed you that we published. We have a working hypothesis on what's happening, and it's happening probably through the fast ligand pathway. Stabilizing the fast ligand pathway helps to reinstate the cell death, through which is induced through either oxaliplatin or gemcitabine treatment. So just to summarize, in AIM-1, the antibody was generated. There was good selectivity and affinity. There was increased cell death with the treatment of the antibody for PDAC cells. And there was impaired ability to migrate through transverse spores and close an artificial wound. And most importantly, we enhanced drug efficacy when you were co-treating it with the antibody. So that brings us to the other tumor type that I talked, and this is pancreatic neuroendocrine tumors. So these tumors are also extremely sensitive to antibody treatment. They also start to die off really fast. And when you treat neuroendocrine tumor mice, these are genetic mice called RIP1 TAC2 mice. When you treat them at particular stage-specific progression, or particular stages that we know in the stage-specific progression, we see different effects. So in the late-stage treatment, which is in the mouse age between 7 to 12 weeks or 12 to 14 weeks, when you intervene in this window, you see that the macroscopic tumors on average get smaller. So what you see here is a single mice, tumors derived from multifocal tumors of a single mice. And basically we average the full volume, and this is a representative image, and we have, of course, large mice numbers when we consider tumor volume calculations. And in early stage treatment, we realized that the number of islets that transform themselves post-angiogenesis into angiogenic islets are significantly reducing with treatment of GSM 192. When we thought about the mechanism, we dug deeper and we tried to look if proliferation is affected. We saw that there was no effect in Ki67. We tried to see if caspase-3, which is an indicator for apoptosis, is affected. We saw there was no significant difference there. We didn't see a significant difference in CD31 levels. These are markers for established endothelial cells, means established blood vessels. But we did see a significant difference in CD34 levels, and CD34 positive cells are angiogenic progenitor cells. These are required for neoangiogenesis. So we understood that MMP7 is really playing an important role in PADNET angiogenesis. We confirmed this ex vivo with assays. There's a tube formation assay using human umbilical cord cells called HUVEC cells. When you do the treatment, you see that there's a reduction in the number of branch points and a reduction in the number of closed loops that are formed by HUVEC cells in the presence or absence of the antibody, GSM 192. There's another assay where we take out the thoracic aorta of mice and we embed it within collagen gel and provide it factors for it to sprout. And in the presence or absence of the antibody, you see here the effects. The sprouting was drastically reduced in the presence of anti-MMP7 GSM 192 compared to the control antibody. So there are a number of known substrates, but there are also a number of unknown substrates that we wanted to look into to complete the whole picture. But this study then had to be done using something called digradomics. Digradomics is basically proteomics of the digradome. You look at the proteome, which includes all the proteases, but also the associated substrates and signaling molecules, etc. And so together, the inhibitors, the proteases, and substrates together form the digradome, and studying this using MSM digradomics. I'm unfortunately not able to show you the results from here on because it's first of all not published, and the reason it's not published is we are trying to patent the second half of this project separately, and until the patent is filed, I'm not allowed to disclose the details of the project. So there is a very interesting story with a candidate from now on, but that story needs to be revealed only when the time is right. So in summary, what I've shown you so far in AIM2, we saw that the treatment really reduced angiogenesis in vivo and ex vivo, and it affected tumor size. And it looks like anti-MMP7 could be even a standalone treatment in a disease like pancreatic neuroendocrine tumor, which unlike PDAC is slightly less aggressive and is also an orphan disease under the FDA category. So this is a patent that we already filed for the antibody, for anti-MMP7 antibody, and all the details can be found here if you go to Google Patents and search for it. I want to thank my lab. This is Professor Irit Sagi, and this is the wonderful team that I worked with for many years, and former lab members who kickstarted the project but left very early on. So I took on the mantle from them. And of course, our collaborators from the Weizmann are also from Sloan Kettering Cancer Center in New York. And there are many funding agencies that I haven't mentioned here, whom we are really thankful for making these projects happen. Presently, I am a postdoc in Tammy Geiger lab. The Geiger lab at the Weizmann looks at clinical cancer proteomics. So because I did digodomics and I was really interested in proteomics in general, I decided to shift to this lab to pursue my postdoc. We are now in the process of onboarding latest technology in MSMS proteomics that will enable us to do studies on cancer heterogeneity in the clinical samples, to perform single cell proteomics, just like single cell RNA. Also, we look into cancer proteogenomics. So it's a fusion between proteomics and genomics. So at the later stage, when my work progresses, I'll be in a position to talk further about the work that's happening now in my postdoc that I joined 6 months back. I finished the scientific talk here, and with your permission, I want to quickly run through my journey so far so you understand the path that I took and my personal experience. I'm very much a Josephite in my DNA. I'm a product of the microbiology department. I was in the MCZ batch, and this is a picture from our whole microbiology batch that we did the bachelor's or BSc there. And you can see your Ria ma'am really hard at work in the microscope here. This was a picture taken during the Christmas Carol competition. And there are fond memories of Joseph's that we still cherish. And it's very important for me to mention that I found my life partner while studying at Joseph's. So Anjana, who also did her PhD at the Weizmann, is now in Israel and is now a postdoc in Weizmann. So Anjana Shanoy Katpadi, I met her in Joseph's. Yeah, that's another picture. And this is your next speaker, Nivedita. Yeah. So from bachelor's in Joseph's, I human genetics and molecular biology at Manipal, Manipal School of Life Sciences and Manipal University in Manipal near Udupi. So I was really enriched by my experience there, and it's actually my director of Dr. Satyamurthy who told me about an opportunity to apply for a fellowship for 8 months in Israel. And since I was doing applications at the stage, I decided to take it up. I did not think ever of coming to Israel. It's not the first priority destination for us Indians. I grew up in Saudi Arabia, so even more so, it was always looked at the other end of the other side of the proverbial fence. Things have changed pretty much already in the Middle East, but but I'm really glad I made the trip to Israel and decided to stay here for my PhD because I earned my PhD at the Weizmann Institute. I came in the beginning to Tel Aviv Sourasky Medical Center in Tel Aviv and then shifted to the Weizmann for my PhD. Weizmann is a really special place. A special place compared to all other institutions in the world as well. It's consistently ranked as one of the best places to work for academicians, for scientists. And it's also ranked one of the top 10 in terms of scientific output. In university rankings, it doesn't do good because it's not a university, it's actually a postgraduate institute. It was established in 1934. It was renamed in 1949 after the first president of Israel, Dr. Chaim Weizmann. And you can see the snippet of information, the impressive numbers that add up to this colossus of an institution that is Weizmann. I earned my PhD in 2020. These are just pictures to show you how cosmopolitan the Weizmann is. It has, as it is written here, many nationalities. Is it mentioned here? No. I think we have around 60 nationalities on campus. It has everything within its four walls, many disciplines and not just biological disciplines. It has very advanced technology, microscopes, instruments. You mentioned it, it's there. You're welcome to check out the YouTube channel of Weizmann Institute of Science to know more about the institution. But I must say that studying at the Weizmann really opened vistas, not only in terms of thinking for me, but also in terms of opportunities for me. I thank you for your attention and I'm open to questions.
Thank you, sir, for that really wonderful talk. Like sir said, the floor is now open for questions. Please post all your queries in the chat box. So we have a few questions, sir. The first one being from Dr. Nivedita. I'll be asking you the question, sir, if that is fine. The first question is from Dr. Poppy Datta. She asks, what is the survival rate of pancreatic cancer?
So it's a loaded question because it depends what survival rate you're looking at. If you're looking at median survival, I don't know, do you see this slide? You see here, it's in months. And in disease-specific survival, also it's in months. So after, I told you, it's mostly detected really late in third or the fourth stage because it's a silent tumor. It progresses really silently without any outward expression of its existence. And by the time it's detected, it's too late. So it's already metastasized to the liver and to other tropic locations that it likes to metastasize to. And hence it's a lost gain when it's mostly detected in the late stage. And then people get from few months, I'm talking about 3 or 6 months, to maybe a year or 2 years, 3 years, but that's usually the range. And treatment doesn't really help because once metastasis has happened, then treatment outcome is also anyway poor. I hope I answered your question, Dr. Datta.
Thank you, sir, for your response. The second one comes from Dr. Jyoti Williams. She asks, is this cancer predominant in men or women, or is there any correlation with age?
That's a good question. There is no correlation with sex or gender. There's no correlation between occurrence of pancreatic cancer and being a female or male. There is a strong correlation with age. Yes, it's actually a cancer that occurs to most patients in their older age. So, because it takes, I showed here that it takes several years for the progression to happen, and most of these events remain occult for some time, and then it takes 12 years. So by the time it's detected, it's already in old age and too late to treat. Yes. I hope I answered you. We can move on to the next question, right?
Thank you, sir. The third one comes from Dr. Beatrice Sequeira. She asks if we can look for any genetic markers like breast cancer. There was a BRC22 that you had shown. Are there any similar markers?
You mean a diagnostic marker? I'm guessing a diagnostic marker for PDAC, or a prognostic marker for PDAC does not exist. PDAC is really, there's an unmet medical need to come up with good diagnostic markers, as well as prognostic markers. What is used in the clinic now are CA19-9 and few others, but they are really not doing their job really well. So as a result, we don't have like a blood-based biomarker, and I haven't shown my results from the blood-based biomarker study here, but one of the other studies we pursued with doctors from Sloan Kettering Cancer Center in New York is to look at blood plasma of patients, especially at the exosomes, and to come out with new targets for biomarker studies. And we have a few targets from the MMP compartment, MMP enzymes. And actually, we are right now communicating a paper on this. So I hope to see it through the review process, so it will be available to you all. But yes, we are really not in a good place when it comes to having a good biomarker, either for diagnostic or prognostic purposes in pancreatic cancer. Some of your research in the future
Thank you.
might be helpful.
Thank you for that, sir. The next one comes from Amrita Nambiar. She says a few years ago,
I think I'm losing you.
There was a high school student called Jack.
Jack
and Raka, who invented a test that would mesothelin antibodies in a nano. Now,
Simran, if you don't mind, I can see the question here. I lost you in between once, so I'll just read the question once again and I'll answer it if you don't mind. So a few years ago, there was a high school student, Jack and Raka, who invented a test that would detect early stages of pancreatic cancer by using mesothelin antibodies in a nanotube. Is this still being used today? How has it developed? So I'm not aware of how exactly I heard about this, and I'm not aware how it developed, if it really was successful in coming to the bedside from the bench. I know that there is a lot of focus nowadays, and also funding nowadays, on coming up with good diagnostic markers, methods like the nanotubes, etc. I am aware that this method is not being used in the clinic widely yet. So as far as I know, this method might be in developmental stages, but it's not being widely used in the clinic. You should understand that in a lot of cancer therapies, COVID was an eye-opener, because, when we talk about time scale for developing antibodies, etc., it used to be in many years, if not in decades. And COVID showed us that much of it was lack of investments, interests, red tape, and we have shown ourselves that we are capable of moving things along much faster when need be, in terms of regulatory approvals and necessary studies. So I hope there'll be increased impetus also for both diagnostic as well as therapeutic applications of biologics in cancer as we go forward. Simran, do you want to read the next question for me, please?
Yes, sir. The next one again comes from Dr. Beatrice. She asks if there is any knowledge about 3D printing of pancreas and transplants.
Yeah, so actually it's a good question from Beatrice ma'am, because we do have a unit at the Weizmann that is really now blossoming with 3D printing. And with many institutions in the US and Europe, there is a lot of push for 3D printing, because we are now in this era where moving on from 2D cell culture, and we want systems in 3D. So of course there was the organoid craze that is now slowly starting to pass, and now we have the 3D cultures with blood vascular nanotubes and supply, etc. Especially about the pancreas, I think I read a paper from 3D printing on the pancreas recently. So yes, I think the field is only now, blossoming, but it's only a matter of few years before we start seeing a lot of studies based on 3D printed pancreas. But some of the focus of the studies in pancreas initially, as always, based on market capital for pharmaceuticals, is in diabetes and study of beta cells and insulin resistance or lack of insulin production, etc., in diabetes. So I'm guessing any new craze in the field, including 3D printing, will be used simultaneously for that as well as for cancer studies.
Thank you, sir. The next one is from Ria ma'am. She asks if there are any symptoms to look out for to detect pancreatic cancer in its early stages.
Most of us are accustomed to ignore small complaints of our body. So I always, when people ask me this, no, there are no acute symptoms to look out for. That's why it's a silent tumor. But there are indicators that you can catch earlier than later. So if you have consistent indigestion, if you have pain in the abdomen that comes and goes, but still, recurs. If you have any other out of the normal in your abdomen, get it checked. I mean, people start with very simple ultrasound, and it's not. And as the age progresses, I really suggest people above the age of 60 to go for regular checkups where they do total abdominal ultrasound. Not only in case of pancreas especially because it's a silent tumor, but in any abdominal solid tumors, and any solid tumors in general, earlier you catch, the better is the treatment available. In terms of pancreas, unlike mammograms in breast cancer and things like this, there is no particular scans, and lesions are always not visible. So this is why we are trying to come up with new diagnostic biomarkers. So hopefully we get there sooner than later.
Thank you, sir. The next one is from Anjali. She asks if this knowledge of pancreatic cancer and its ECM, whether it can be related to any other cancer type, or if the role of MMP7 is dominant in any other cancer apart from pancreatic cancers.
That's an excellent question. And so the antibody we created, we tested it in pancreatic cancer, but does not mean it will only work in pancreatic cancer. It will very much work in other cancer forms where MMP7 is known to be playing an important role. So colorectal cancer, CRC, is another cancer where MMP7 plays an important role. And when you look at mRNA expression data from many cancer types, you know that many cancers of the gastrointestinal tract has high expression and activity of MMP7. In addition to that, MMP7 was initially discovered in the uterus. So in endometrial cancers, also MMP7 is high. So what I didn't tell you is that after the patenting process was tabled, we are now in the process of working with, through the technology transfer arm called YEDA of the Weizmann Institute of Science. We are in the process of working with venture capital firms to try and get funding to test it and commercialize this antibody. So hopefully we'll get there soon.
Thank you, sir. The next one is from Hazel D'Souza. She asks if there are any early warning signs of pancreatic cancer and how serious a tumor on the pancreas is.
Well, I told you there are no real early warning signs for pancreatic ductal adenocarcinoma, but neuroendocrine tumors, because most of them are functional, then because there is hyperinsulinemia or hyperglucagonemia, you get to detect it. So the percentages of pancreatic cancer in total cancer occurrence is not that high, actually 7-10% in the US, where numbers are available. But the percentage of survival, patients who have long-term survival is abysmal in pancreatic cancer. So are there early indicators that we can know? Unfortunately, none. Is there things you can do? Yes, regular checkups post the age of 60, not ignoring symptoms that might come and go and things like this. Get checked up. It's a basic advice that anybody like me could give.
Thank you for that, sir. The next one is from Anusruti Nair. She asks, what are the side effects of the MMP7 inhibitory antibodies, and how is this line of treatment better than the treatment methods that are currently being used?
So MP7, so biologics as treatment for cancer is a field that is still only in the progress of growth. I mean, you have, checkpoint inhibitors that are used against PD-1, PD-L1. These are already commercialized. MMPs as targets did face a big setback few years back because there was in clinical trials a small molecule inhibitor against MMP9, which failed miserably. Like it was in a bad state in terms of the outcome of the clinical trial. It was a badly designed trial, and there were many flaws to it, to the preclinical studies that was associated with this clinical trial. So we are looking at it in a completely new perspective. We are not creating small molecule inhibitors which have less selectivity and less potency. We are looking at creating biologics like antibodies, which have very high affinity and very high specificity. Also, the antibody against MP7, GSM192, it binds to the active enzyme only and not to the proenzyme. This is very helpful in terms of MMP targeting. But the specific question of what are the associated toxicities, we will only get to know when we start to do advanced trials and do, mouse necropsies and then go into human trials. So toxicity is the first thing that we look at in trials. And from the indications that I have so far in our treatments, it's not a toxic antibody. It doesn't have systemic effects. It doesn't have any major noticeable side effects. But it may not really be a good standalone treatment in PDAC because it's a very aggressive tumor, but it could be, as I showed you, a co-treatment agent that reduces the amount of chemotherapeutics that is required to get good cell kill. And in neuroendocrine tumors, perhaps it could be a good standalone treatment. But whatever I showed you was in mouse. There is an ocean of difference between mouse and human. And this is why we have clinical trials. And this is the path on which we are. So we will test it out.
Thank you, sir. That was very comprehensive. Dr. Beatrice has another question. She asked, can the MAB raised be used as a drug?
Yeah, so the MAB, now that I showed you the monoclonal antibody, is basically a mouse antibody against a human enzyme, because the enzyme is very well conserved. So we used a human target to raise it. So it targets and binds and stops the activity of human enzyme. Now, in the next process, when the interested parties like the venture capital firm or whoever they assign takes it, they have to humanize the antibody. So there's a process in which the mouse FC is replaced with human FC. So you humanize the antibody, and then it can be used for clinical trials to develop it as a drug. So yes, we are hopeful that the antibody continues to show the good effect that it shows. It can get humanized, and also it can be synthetically produced and not from hybridoma. And these are things that we are pursuing right now. Thank you.
Thank you so much for that, sir. If there are any more questions, please do post them in the chat box. If there are none, we will close the Q&A session. All right, sir. Thank you so much for the talk. We learned.
I just want to add something
please,
before Nivi takes over. When we were in Joseph's, I'm sure Nivi feels the same. Nivedita feels the same. We couldn't map it all out on what we are going to do, where we are going to do, how we are going to do, and that's perfectly okay. I think you are getting good training at Joseph's to start with, and I hope none of you get disheartened with 1 or 2 rejections. Keep at it, and I'm sure many of you will follow our path and come to academia and research. Thank you.
Thank you so much for that, sir.
Hi, Nivedita.
Can you hear me?
You're on mute. Yeah.
Hi. Hello, ma'am. So nice to see all of you. Oh my God. It was so lovely to see everybody after so long.
It is.
Yeah, it is.
As Vishnu was amazing. It was an amazing talk. I caught the last part of it, but it was really good.
Actually, all your pictures, old memories, no? So that again brought us back to your lab durations when you all sit together as a group, writing the records, hiding in the next lab and writing all.
Thank God he didn't have the picture of that.
Yeah.
Actually, I was looking through all the pictures the other day, and I found some of those, like all of us hiding using the cell phone under the bench.
Really, Nivi? I'm not your lab, ma'am. Not your lab.
Not your lab, yes.
They're not in your lab.
None of them are in your lab. The pictures are from
I was remembering how we had to wait for the tea person to come in.
Yes.
And get us all tea. And I think those were the best days, most carefree and the best days.
Sure.
It also tickled me when Vishnu said Ria ma'am. I was like, I can't imagine Ria as Ria ma'am.
It sounded very weird coming from Vishnu.
This timing actually works out perfectly. It's 6:30 in the morning, so it's peaceful and quiet in the morning here. So it worked out just fine.
Great. It works out for us because all the classes are done. So UG students are free from their classes. PG students, we can, reschedule theirs. But UG students have different classes. So that's why we usually keep it post 4. Through the day, there are other stuff.
Are all the classes still virtual now?
They are now this month. We were offline till beginning of Jan, but then with the cases riveting up the sky, so we went virtual again. I think it's going to stay for like that for some time. The decision is pending.
Yeah.
Vishnu, all the best for your patenting.
Thank you.
Every time you talk about MAB, I get so excited.
I learned the basics from you.
Oh, that's okay. I always tell you, start a firm.
It was a great talk. Great one.
Exciting.
Thank you. Thank you.
How's the little one, Vishnu?
She's
keeping you
great. I mean, because the cases are really high here, we have a lot of disruption in the childcare system.
right.
So it's quarantine, not quarantine, in, out, days of work that we plan, and then suddenly we cannot do because she cannot go. Yeah.
I know. Oh. How old is she now? 6,
She's
8?
7 months.
7 months.
7.
Okay. So is she keeping you up at night? Does she sleep through the night? Okay.
Sleep is still an issue, and we are trying to tackle. Yes. But otherwise, she's very cooperative on food and all other things, but sleep is still an issue.
So sweet. Yeah.
Simran, can you just go ahead?
Yes, ma'am. Sure. Thank you so much, sir, for your talk. We have reached at the end of the session. I would like to take this opportunity to extend my heartfelt thanks to Dr. Vishnu Mohan for taking time out from his very busy schedule and talking to us here at Kaleidoscope 4.0. We not only learned from your talk, sir, but we also learned a lot from your personal journey that you shared with us. I would also like to extend my gratitude towards my team who helped me go through the Q&A session very smoothly, and to the entire microbiology department who has worked tirelessly to make this session a very good success. Lastly, I would also like to thank the audience who has been brilliant throughout. I take your leave now. A feedback form has been posted in the chat box. Please make sure to fill it. The next session will start in a while. It will be moderated by Yogada, and we hope to see everyone here again. I now hand over the floor to Yogada. Yogada, you can take over now.
Thank you, Simran. Good evening, everyone, and a warm welcome to one and all present here. I'm Yogada Panse, a student of first year MSc Microbiology, and I will be a moderator for today. I welcome you all to the third session of Kaleidoscope 4.0. I request all the participants to keep their microphones on mute and videos off during the course of the session. I also request all to not leave the meeting abruptly. Kindly put your questions in the chat box and we will present them to the speaker after the culmination of the talk. Our today's topic is harnessing the power of your immune system to fight solid cancers. It is a pleasure to have Dr. Nivedita Mohan Ratnam as a speaker for this session. She is currently pursuing her postdoctoral fellowship at the surgery branch, NCI, Bethesda, Maryland, USA. Her area of research is in manufacturing of genetically modified and non-modified lymphocytes for adoptive transfer in patients with metastatic cancers. Her prime interest lies in understanding the process of tumor development and cancer-related research. She is an alumna of St. Joseph's College. She completed her bachelor's in microbiology, chemistry, and zoology in the year 2009 and went on to pursue her master's in microbiology in the same college of batch 2011. She completed her PhD at the Department of Molecular, Cellular, and Developmental Biology at the Ohio State University, USA in the year 2017. She is skilled in handling techniques in various fields like molecular biology, cell biology, patient tissue processing, to name a few. During the course of her career, she received many grants and has many awards in her name. Dr. Ratnam has also published numerous research articles in reputed journals. She has been a mentor and role model for several students. It is an honor to have you with us, ma'am. Over to you.
Okay, can you see my screen? Okay.
Yes.
Thank you for that very kind introduction. And good evening to all of you. And I know this is the last talk for the day. So I won't be presenting much data, but what I hope to tell you is a little story on the development and current state of immunotherapy for cancer. And what I hope to sell to you is the benefits of cancer immunotherapies in combination with what we know as traditional therapies, which includes chemoradiation therapy in my talk, which I've titled Harnessing the Power of Your Immune System to Fight Cancer. So I call cancer the 800-pound gorilla. It's here to stay and it won't go until we do something about it. And data from the World Health Organization as of 2020 ranks cancer as one of the top 5 causes of death globally. And as you can see, I don't think there's any place that has been spared, including in India, where it is among the top 3 causes of death. So what is cancer? It is not one disease, but a collection of over 1,000 genetic diseases, which ultimately results in the uncontrolled proliferation of cells. And how does a normal functioning cell go from a normal cell to becoming a cancer cell? So on an everyday basis, we as humans are subject to various environmental and genotoxic stresses. And what happens is as our cells divide, they accumulate mutations. And over a period of time, if the accumulating mutations affect certain driver genes and certain passenger genes that cause replicative stress, then what happens, which sometimes is irreversible, then that leads to the formation of a tumor. And so broadly, from the time that a normal cell progresses to a tumor cell, you can roughly divide it, and there's no clear demarcation, but roughly divide it into 3 stages: initiation, promotion, and progression. And there are certain hallmark characteristics of tumor cells, as you can see in the figure on the left. One is it has an inflammatory microenvironment. That means it is filled with a lot of immune cells that are trying to destroy it, sometimes successfully, most times not. It is insensitive to growth inhibitors. It is self-sufficient to promote its own growth. It has a limitless replicative potential. That means it can go on dividing. It has sustained angiogenesis. So it has its own network of rapidly dividing blood vessels that can bring in nutrients and the required sustenance factors for the tumor cells. It evades apoptosis, that is, it avoids cell death. And it has the ability to invade and metastasize, that is, spread to different parts of the body, no matter where its origin is. So one of the thought-provoking points in all of this is this entire process that I just described to you, and as Vishnu mentioned in his talk, takes place over a long period of time. It doesn't happen in an instant. It's not like today, a cell began to accumulate mutations sufficient to drive it towards tumorigenesis, and tomorrow you have a tumor. It takes years. And it's not just this process, but there is other factors that contribute to this delay in clinical presentation of tumors. And as a result, when you look at the occurrence of cancers or tumors in general over age, you see that there is a susceptible range of people who present with tumors. And usually that is people over 50 years of age. Now, why does this happen? So there are 2 mechanisms that can be attributed to this late presentation of tumors, as well as the association with age. The first is what I've shown to your left, which is intrinsic tumor suppression. Now, like I said, through our lifetime, we are exposed to a lot of genotoxic stress. So when our cells divide, they accumulate mutations, and some of them are oncogenic. However, the cell's internal replication machinery has the ability to recognize some of them and correct some of them. But once the mutational burden reaches a certain critical threshold, what happens is a program of cell senescence, that is a stage where the cells neither die nor multiply. They're basically kind of dormant, but more towards the dying cells stage, is triggered. And that kind of prevents the cells from just going through this limitless replication and becoming cancerous. However, when there is an inactivation of the tumor suppressor genes, then this senescence program is pretty much awry. And then that leads to the uncontrolled proliferation of these tumor cells. Now, the other aspect of it is cell extrinsic. It is an extrinsic tumor suppression mechanism that we have in our bodies that we can attribute to the immune system. So what happens is if you think about it from an age trajectory, very young children and older people have very weak immune systems. So it is kind of natural that they are more susceptible to getting disease. Take COVID, for example. Mostly affects very older people, younger people. It does affect people in the healthy range between, say, 20 to 50. But if you are, say, vaccinated, your symptoms are much lesser. You don't suffer for as long. So that's because we have a healthy, competent immune system. The same holds good in the case of cancers as well. So what happens is when a normal tissue escapes this intrinsic tumor suppression mechanism, it starts expressing certain proteins that are not always native to a healthy cell. Excuse me. That are not always native to a healthy cell. And these are danger signals that are picked up by the immune system. And the cells of our immune system, both innate and adaptive, in sequence, they move to that site where they are detecting or sensing these danger signals in an attempt to eliminate the tumor cells. So this is the phase where they successfully come in, they see the danger signals, they eliminate them. However, tumor cells are smart in themselves. So what they do is they go into a state of tumor dormancy and editing. So what that means is that they kind of stop expressing these danger signals and they start understanding what these immune cells like, what they need to thrive and survive, what they need to kill these tumor cells. And they almost, you can say, just try to reverse engineer their genetics to make it so that these immune cells actually like them and not hate them. So that is a stage of tumor dormancy, which is predominantly during the late, early adult stages in human life. And then you have this third and final stage where the tumor cells gain enough control over the immune system that they are able to overpower the immune system. This is called the phase of tumor escape. And it is potentially at this time that the tumor cells really are able to grow and spread, and when they start showing clinical presentation. And this couples very well with the later ages in life, where the older adults have a weakened immune system. So this entire concept is called the theory of cancer immunoediting. So before we go into more details, and I'm sure Beatrice ma'am covers this in good detail in class, like she did for us, I just briefly go through the key players in the immune system. So as we know, we have two arms in the immune system, the innate immune system and the adaptive immune system. The innate immune system is usually the first line of defense. They are known to sense danger signals the fastest. They are like the first responders for any natural calamity. So the main cells include basophils, eosinophils, mast cells, natural killer cells. And during the phase of elimination, it's mostly these cells that go into the site of tumor development and try to clear out the developing tumor cells. Then you have one arm of the couple members of the innate immune system, such as the macrophages and the dendritic cells, which in addition to being part of innate immunity and being able to secrete cytotoxic factors that can kill infectious cells or tumor cells, they also have the ability to communicate to the adaptive immune system, which is the second part or the second phase of our immune system, which is mostly more long-lasting, but kicks in much later than the innate immune system. These are the macrophages and the dendritic cells. So what they do is when they kill the tumor cells, they process some of the proteins that are expressed in these tumor cells and present them to the T cells and the B cells so that the T cells can make cytotoxic cytokines that can kill the tumor cells, or the B cells can make antibodies that are able to neutralize these cancer cells. So with that understanding, like I said, the adaptive immune system provides immunity that is more long lasting. And that is what is required in the prevention of tumor development. I'm going to focus a little more on the T cells, which is where my group's expertise lies. And so to understand exactly what we do, I would like to spend a few minutes trying to explain the mechanism of action of T cells. So T cells generally follow what we call a 2-signal or a 3-signal hypothesis in order to be activated and carry out their function. The first signal is when these antigen-presenting cells, the macrophages or the dendritic cells, present the protein that they have digested and processed from the tumor cells on their surface. So that's the first signal. And this is recognized by a specific T cell receptor that is present on the surface of the T cell. The second is co-stimulation. This is a second round of signals that the antigen-presenting cell provides to the T cell for it to say, yes, you have been engaged and you have been called on to destroy cells that are expressing this particular protein that we have shown you. And the third signal is called the instructive cytokines. So these cytokines include are secreted from the antigen-presenting cells, tell the dendritic cells what to differentiate into. So you have a naive cell that has seen a antigen on the surface of an antigen-presenting cell. It has received co-stimulation. Now the cytokine tells it differentiate towards an effector response because we need response right now versus we need memory response. We need you to keep in mind that if you see something like this, you need to be activated in the future and you need to destroy the cell. So these cytokines are more instructive in helping them differentiate. And where does all this action happen? So it happens at 2 different places. The first and the primary stimulus takes place in the lymph nodes. So once the antigens are processed by or the cancerous cells are processed by these antigen-presenting cells, they bring these antigens to the lymph node. And as we know, the lymph node is the site where T cells are generated. So it brings it to the lymph node, and then it provides the first round of stimulation to the T cells. From there, the T cells migrate to the site of the tumor, where they're able to get a second dose of stimulation because the tumor cells express those proteins. So they get a second boost. And that is how they're able to successfully kill the tumor cells. However, nature has devised our immune system in such a way that for every positive action, there is a negative reaction to control that positive action. So imagine you have an infection or you have a cut. Let's take it that you have a cut on your hand. It's infected. Your T cells come in They clear the infection. Now, what happens if they don't know how to stop? If they don't know how to stop, chronically, your wound is never going to heal. You're always going to have pain. There's always going to be pus oozing from it. So the immune system is designed to have its own breaks. Now, what happens is, in the case of tumors, they are very smart. I told you in that equilibrium phase, the tumor cells are kind of surveying and understanding what these immune cells like and don't like, what they need for functioning and what they don't. So what they do is they learn that these breaks are very interesting. How we can gain control over the immune cells is if we hijack these breaks. So if we make sure that along with expressing the antigen proteins, we express a ton of these brake molecules, then the immune cells get confused because they're getting both a positive signal and a negative signal. They generally are rendered ineffective. So there are two major breaks that I'm going to tell you about. There are several brake systems, but I'm going to talk to you about two important ones. One is called CTLA4, which happens very early in the stimulation of the T cell within the lymph node, where expression of CTLA4 causes a negative feedback to the T cell and it remains ineffective. The other is at the site of the tumor, where the tumor cells express something called PD-1. The tumor cells express the ligand PD-L1 that binds to PD-1 receptors on the T cell and gives them that negative signal. So one of the ways in which people have therapeutically targeted this interaction is by making antibodies against CTLA4 and against PD-1. So what we do is we can give antibodies to patients that can help negate this interaction between CTLA4 and the PD-PDL1 interaction. So that's one way of doing it, but that's proven to be insufficient as a monotherapy for most cancers, although it is commercially approved for a number of them. So what are the main strategies of immunotherapy that are currently in research, many of which are in the clinic as well? One is cellular therapy. So one is cellular therapy involves trying to engineer your own T cells to make sure that they are functional. They move from that non-functional state in which your tumor microenvironment has made sure they exist. You try to reprogram them and make them active against the tumor. The other is immune modulators, which I just spoke about, which includes mostly inhibitors against these checkpoint blockade molecules like PD-1 and CTLA4. The third is oncolytic virus therapy. So that's a very interesting strategy where you have modified oncolytic viruses that can kill cells, like the HIV virus, that have been modified in the lab so that they don't have the capacity to be bad for you, but they can recognize and kill the tumor cells. So that's an ongoing research. The next is monoclonal antibodies. These are antibodies that attack certain specific parts of cancer cells. And Vishnu spoke a lot about it to you, so I won't go into detail. And then you have cancer vaccines. So in this case, like most times we recognize vaccines as mostly being preventative, but unlike the preventative vaccines, in cancers, we use prophylactic vaccines. So that means that we inject sequences of, say, mRNA that are native to the tumor cells that the immune cells can take up, process, and attack the immune cells. And interestingly, some of the RNA-based vaccine therapies that are currently being administered from Pfizer and Pfizer-BioNTech and Moderna actually stem from research that was done in cancers. And it is because of that accumulated knowledge that we had going on with cancer treatments and trying to use mRNA vaccines to treat cancer patients that we were able to mobilize and come up with vaccines against COVID-19. So that's an interesting bit of information. And my group is focused mainly on cellular therapies. And that is, we try to modify the patient's immune cells so to make them, or to reverse their functional inertness, to make them active and try and attack their tumors and help clear those tumors. So the one of the ways in which we do it is adoptive transfer of autologous T cells. So that means what we do is when a patient is identified as someone who can come onto our study, then we do a tumor resection. So the surgeons resect the tumor, and then we get these tumors in the lab. What we do is then we chop up that tumor into many small bits. Typically, a single tumor is usually cut up into 24 small bits. And what we do it is we add media to them. So we give them nutrients and we put them in a 24-well culture dish. And we add IL-2, which is a T cell growth stimulating cytokine. And then we just wait for days. Some of these fragments will allow T cells that are present within that tumor fragment to grow out. Some of them will not. So you select for the ones where we are able to get T cells from that grew from that portion of the tumor out, and then you assay those tumors for recognition against tumor-specific antigens. So what we do is that we take these T cells, we culture them with some of the tumor cells and see if they're able to kill the tumors. Now, not all T cells that are found within the tumor are actually tumor-specific or actually have the capability to recognize the tumor. Some of them are so far gone that they don't have the capacity anymore to recognize these tumors. So then we select for those that can recognize the tumor cell and successfully kill them in the dish, and then expand that. So we do what is called as a rapid expansion protocol. So essentially we go from a few thousand cells to almost 10 million cells. That's how much we need to expand. And so we bombard it with a bunch of growth cytokines, provide them all the best nutrients, and we provide them accompanying feeder cells, which are basically irradiated lymphocytes and everything that they need to grow rapidly. And then we reinfuse those into patients after the patient has gone through a dose of lymphodepleting chemotherapy. Now, why we do that is because we want to make sure that the cells that we are infusing into the patient has a chance to grow and expand rather than be overrid by fresh cells that are made within the patient's own bone marrow. So you give them lymphodepleting chemotherapy first for a few weeks, and then for 2 weeks, and then after that you administer them with the selected tumor infiltrating lymphocytes. And so far, this, the success rate, although not astronomically good, has been pretty good. We have a decent success rate, especially with a lot of melanomas and some liquid cancers as well. Now, then there is another way in which we kind of tamper with the patient's T cells to try to see if we can get them to function better and recognize the tumor better. So once a patient is identified as a suitable candidate for our study, what we do is we resect the tumor. And this time we divide the tumor into fragments from which we don't take out the T cells, but we extract DNA from these tumor tissues. What we also do is we take some amount of patient peripheral blood, and from the patient peripheral blood, what we do is we take some of the T cells, and we also take some cells just for DNA extraction. And what we do is we do what is known as whole exome sequencing. So we look at the DNA sequence of the tumor cells. We look at the DNA sequence of the normal cells, and then we use algorithms to detect what mutations are not seen in normal cells, but they are seen in the tumor cells. And once we do that, we try to identify what T cells can recognize this specific mutation that is seen in the tumor cell, which we call the neoantigen. And then we genetically engineer the patient's T cells that we got from their peripheral blood to express these particular T cells, to express these particular TCRs or T cell receptors that can recognize neoantigenic mutations. And then we expand them. We use selection markers to make sure that these T cells are activated. And then we reinfuse them into the patient's back again, and then see how well these engineered T cells can kill the tumor cells. So this is a more, an in-depth view of how this is done. Now, in the same way as how we engineer the patient's autologous T cells taken from peripheral blood to express T cell receptors against neoantigens, there is another method, which we call chimeric antigen receptors, which are basically genetically engineered T cell receptors that are high affinity, that have known to function with greater efficiency, that we can engineer onto the T cells of the patients to successfully target the tumor cells. So these chimeric antigen receptors are slightly different in structure than native T cell receptors. So they are basically engineered in the lab to have the best parts of a T cell receptor and the best parts of an antibody response, and kind of combine them. And it's a genetically engineered chimeric construct. And then it's the same process. You expand these cells, then you precondition the patients with lymphodepleting chemotherapy, and then you give this T cell product to them. So a huge success story in the world of chimeric T cell antigens is the FDA providing approval to use CD19 CARs against AML patients, which is huge, because this is one of the first times that a genetically engineered T cell treatment has been approved by the FDA for as first line treatment for patients. So just walking you down the history of the evolution of immunotherapy, the first treatments really started in 1950s, the late 1950s. And as you can see, that with the increase in knowledge that we actually got with understanding the nitty-gritties of how the immune system works, dramatic progress was made, especially between the early 2000s to where we are now, it has been a complete 180 in terms of really not knowing how to treat this disease to the numerous options that we have right now. And the advent of the CRISPR-Cas9 genome editing technology has also brought about great impetus to the process of moving forward and trying to devise novel therapies for the treatment of cancer. So so what the reason I put this timeline up is because it's hard sometimes to appreciate how much progress science has made when you see around you that patients are still dying from cancer. But if you look at this timeline and see the number of options for treatment out there are a lot more than they used to be. And there are some cancers like pancreatic and glioblastoma and brain cancers that so far have proved to be far more challenging than many others. But we do have a lot of treatment options that harness the ability of your own immune system to treat these cancers that are doing wonderfully in many other solid tumors. And we hope to get approval for more of these treatments as first-line therapies. So like everything in life, no immunotherapy regimen so far is perfect. They all have their limitations. So with adoptive cell transfer and using the tumor-infiltrating lymphocytes to expand and put them back in the patients, one is production failure. So sometimes if a patient's tumor is a bit too far gone by the time they come to us, and we're not able to get enough numbers of tumor-infiltrating lymphocytes from their resected tumor, or the T cells within those tumors really don't show any reactivity, then that is pretty disappointing because we're very limited in how much we can expand and give these patients, because while there is no golden number to exactly how many or what order of magnitude cells these patients need to receive, too few is too little. So we need to be able to give them at least 5 or 6 million cells minimum, and that can sometimes be a challenge. And then as far as the T cell receptor technology goes, there are two main potential limitations for this therapy. One is MHC restriction. So it is very difficult to use these as off-shelf treatments. These are indeed only designed for personalized therapy unless we are looking at a shared antigen because of MHC restriction. That is, every one of humans express a certain combination of MHC molecules that recognize certain antigens, and this is not shared between people. So it's kind of unique to an individual, and so that can be an issue. The second issue is when you are trying to engineer T cells to express the TCR of your T cell receptor of your choice, it is important that the alpha and beta chains, these are the two components of the T cell receptor, they pair correctly, because if they don't pair correctly, they're not going to recognize the tumor antigen correctly. So appropriate pairing is important. And sometimes that can be a problem. However, one way around this problem is using a mouse form of the consonant region so that they pair together correctly. The third is with the chimeric antigen therapy. One of the major limitations is it is limited mostly to surface antigens. So it can recognize only antigens that are expressed on the surface of the tumor cells. The second is, because they are engineered to be so effective, they can result in what is known as a cytokine release syndrome. So what that really is, you have made a T cell that expresses receptors that recognize antigens with such vigor and can activate the T cells so well that you have an excess of cytokines that can kill the T cells, that can kill the tumor cells. And so that excess of the cytokines, what happens is it not only affects the cancer cells, but it can affect the healthy cells also. And it can lead to neurotoxicity. And that is a very major side effect and a limiting factor in these patients. So they have to be observed very closely and given a lot of, treatments on the side to control this. Even then, sometimes the patients have been seen to succumb to these side effects. So right now, the field is in the process of trying to overcome some of these limitations, while also trying to find newer and better ways for making this adopter cell transfer process more efficient. So what are some of the future options? So this is just very broadly what some of the future directions that are being pursued, not just by our group, but in the field, is trying to, for the most part, it is trying to combine different therapies. So one thing that we have learned lessons in is cancer cannot be attacked or cured most of the time with any single agent or a single therapeutic regimen. What we need is a combination of regimens. Now, that doesn't mean we can throw anything on the wall and it will work, which a lot of labs are trying that approach, but it would prove most fruitful if there is a method to the madness. And so kind of trying to devise the timing and combination of treatments that might work best for a given cancer is one of the key focuses in the field to try to improve the efficacy of immunotherapy. The other half is like what Vishnu mentioned in his talk, is identifying biomarkers. So even with the immune system, it is important to identify biomarkers to see how well your immune system is doing. So in order to define the timing of your treatment, you need to have a biomarker for progression. You need to have a biomarker to be able to distinguish an effective response versus an ineffective response, rather than always going the invasive route. So you need blood biomarkers. So that's another field that is kind of, working in parallel with designing newer therapeutic regimens. So with that, I end my scientific talk, and I just like to take a few minutes to describe my journey. I think step one for me was my time in St. Joseph's College in Bangalore, which really laid the foundation for me in basic sciences. I mean, just looking at these pictures of the building brings back so many wonderful memories. I think the teachers, the faculty were fabulous at giving us the basics. And I will tell you for a fact that in comparison to a lot of kids I went to graduate school with in the United States, I had a very sound and a much more in-depth foundation into the science, biological sciences. And I with all my heart, thank the faculty at St. Joseph's for giving us that very sound foundation. And of course, we made friends that have been friends for us now for almost more than a decade. We've been through all our major ups and downs in adult life together, and we still remain as thick as thieves, even though we all live in different time zones almost. So wonderful memories. And then after completing my master's in symbiosis, I took 1 year off to be a research volunteer at IISc and in the Department of Microbiology and Cell Biology. And one of the main reasons I did that was in trying to talk to people in the United States and looking at some of the requirements for applications, etc. I did realize that I was lacking in training on how an independent lab works, which is a little different from demonstrative lab work that we get a lot of in college. And so I felt, rather than being a fish out of water over here, I take the time out to actually see the workings of a lab and how a project is run. And I was fortunate to be able to find a lab at IISc where I could spend a year trying to do that. And it was, the project I was given was a virology-based project, and was lucky to get a second author publication out of my stay there. So that was icing on the cake. Then from there, I moved into basic research, and I did my graduate studies, my PhD at Ohio State University, which is one of the largest public universities in the United States. It is located in Columbus, Ohio. And they are amongst one of the top ranked NCI funded institutions. I did my graduate work in the lab of Dr. Dennis Guttridge, who now who moved his lab now from Ohio State to Medical University of South Carolina. But interestingly, my graduate research was also in pancreatic cancer development. And so much of Vishnu's talk was very interesting to me as well. But I was looking more on the immune side of how pancreatic cancers develop, and that resulted in a couple significant publications that taught me a lot about the process. So it was more basic research in that we never went to bedside with my studies. It was mostly bench to animals. So a lot of studies in mice, a lot of mouse work, a lot of surgeries in mice, a lot of treating mice, a lot of taking care of mice. So that was a fun learning experience. And from my lessons in basic research, I then wanted to see like the process go through, and I wanted to see how does research translate from what we see at the bench to an actual patient bedside. And for that I moved to Washington DC, where I started my postdoc in the neuro-oncology branch at NIH, and worked with Dr. Mark Gilbert, who is a pioneer in the field of neuro-oncology. What I really learned in my time at the NOP was how to design a clinical trial, and also how to analyze data from a clinical trial, how to plan to collect patient samples, what we are looking for, how to come up with tests and biomarkers that we can use to report outputs on clinical trials. And yet while that was one part of it, the other part of my research was also trying to look at coming up with ways to improve immunotherapy outcomes for patients with central nervous system malignancies, mainly glioblastoma. And so the way we looked at questions in translational research is a little different. You ask your questions, or you ask your research questions based on observations that you make in patients. And so that is a very different mindset from basic research, where you ask your research questions based on processes that you see in the lab. And then after having completed my postdoc, in June of 2021, I moved to the surgery branch in the NIH as a research fellow, and I work slightly further down the pipeline now of clinical translational research, where I work, I lead their process development team for manufacturing adoptive cell transfer therapy products. And so my work is mostly trying to identify and scale up research platforms for manufacturing and ultimately for use in patients. And I'm extremely fortunate to be working with Dr. Steven Rosenberg, who is our branch chief, as well as one of the pioneers in the field of cancer immunotherapy. He's one of the first investigators in the world to be able to use immunotherapy to successfully treat patients with cancer, as well as one of the first to have been approved to genetically engineer a human cell for use in the clinic. So it is extremely humbling experience, and I have been fortunate enough to have a bunch of grants and fellowships in the process that have taught me to think independently, including a pre-K99 grant. Yeah, that's me, and that's about it. And I will be happy to answer any questions.
Thank you so much, ma'am, for sharing valuable and interesting information regarding cancers, immunotherapies that are being used to fight these cancers, and the in-depth talk about ACT was certainly an eye-opener for us. Your journey is definitely inspiring. Now, before we open the floor for the discussion, I would also like to remind the participants that the feedback form will be shortly posted in the chat box. So the first question is from Dr. Beatrice Sequeira. If you rapidly try to force the cells to grow, can it also lead to mutations and increase in cancers?
So if this is about the T cells, ma'am, no. So with the rapid expansion protocol, it's actually very short. It's only for 14 days, and it's very controlled. So we don't, but we also have to do quality evaluation before release. So we do take samples to check for their phenotype and functionality before it goes into the patient. So if there is anything that has gone awry in the process, we try to pick it up before it goes in. So I hope that answers the question.
Thank you for your response, ma'am. The second question is also from Dr. Beatrice Sequeira. How long does it take for this method to get so much cells as you need to engineer the expression of neoantigens to inject into the patient?
Right. So usually the entire process, starting from identifying the candidate TCRs, engineering them, going through that process, typically takes at least 6 months in general. Not for the lack of personnel, but just some of the processes take a long time. Just manufacturing them commercially takes a long time. And so a large part of research now is also focused on trying to find ways to shorten this time. And my husband's involved in some of those studies as well. So it takes at least 6 to 9 months. And that's the unfortunate thing, because a lot of the patients that come to us, especially at the NIH, sometimes they don't have that time. And unfortunately, there have been times midway where we have had to, throw our hands up in the air because patient no longer is either a candidate or hasn't made it. But for the most part, that's how long it takes to get to the point where the treatment is ready for patients.
Thank you, ma'am. The next question is from Anjali. How can the T cells injected be ensured to move towards the tumor site only? Is there a mechanism to enhance that process?
So that's a very good question. So there is work going on right now to try to see if we can also engineer clues that drive the T cells faster and more specifically to the tumor. But since they are go through this process of expansion in a culture dish, both the tumor infiltrating lymphocytes as well as the engineered T cells, they are activated, and an activated T cell is programmed to have cues or signals that can help it drive it to the site of the target. But when we inject them into the patients, and we try to aid this process by doing that lymphodepleting chemotherapy in the patients, so that the patients' new and fresh T cells are kind of held back in the bone marrow, and your injected T cells go through. So they technically generally tend to home first to the secondary lymphoid organs like the spleen, and then from there they go back into the tumor. In the natural system, what really happens is, I told you that there is that interaction that happens in the lymph nodes between the antigen presenting cell and the T cell. And there is a third cytokine signaling that tells it to differentiate into a particular phenotype. Part of that third signaling is one of the several cytokines that it gets through that is usually signals to home to the correct tumor site. These include chemokines like CXCL9, CXCL10, CXCL11. So for our studies, we don't really additionally provide those signals, but there is ongoing work trying to see if we need to provide these additional signals either by engineering something on the T cells or just providing cytokine therapy in addition.
Thank you, ma'am. The next question is from Shalmali, ma'am. How do you select the T cells that recognize cancer cells from the tumor mass? Are there any specific markers for the same?
So that's a very good question. So really what we do is a co-culture assay. So we don't really look for specific markers. So when we get the tumor, we cut it up into many fragments. We also keep some of the tumor cells in culture. And what we do is we take those tumor cells and we co-culture them with the T cells. And then we see whether these T cells are able to kill the cancer cells. For that strategy, we don't necessarily look into the details of what antigen, what T cell receptor. Can the T cells in this fraction kill the tumor cells? It's as simple as a plus minus answer. And then we get antigen presenting cells or like dendritic cells that we grow up from the patient's peripheral blood that is part of this co-culture system. And then we look at activation of the T cells. And once we see there is effective killing, we also look for the expression of T cell activation markers like 4-1BB. And interferon gamma and such things to know for sure that these are the T cells we want to take out.
Thank you, ma'am. The next question is from Dr. Beatrice Sequeira. While doing T cell transplantation, will there be a cytokine storm if the concentration is too high?
That is a great question, ma'am. But with these transplanted T cells, what we see, like I said, they first home to the spleen. And then from there to the T cells, to the tumor. A lot of times, many of these, because these are autologous T cells and with autologous TCRs, and they're not the high efficiency TCRs, even almost most of it gets filtered out at the spleen. It's a small part of it that goes into the tumor, but to make sure that that minimal load goes to the tumor, we need to give patients a ton of cells, and that's kind of the catch-22. And also these patients are closely monitored because they can just have regular like side effects, because after you administer patients with the T cells, we give them increasing doses of IL-2, which is the cytokine that helps T cells replicate. So there are some side effects that the patients do have with that, but since they are not those kind of like high efficiency TCRs like in the chimeric antigen receptors, cytokine storm is not really the issue. It is delivering a minimal load to the tumor.
Thank you for the response, ma'am. The next question is from Samson. Cancer relapse occurs in some patients. Do the therapies that you have discussed reduce or even nullify the chances of a relapse?
Sorry, just give me a second. Can you repeat that question, please? Sorry, I didn't get that clearly.
Yeah, sure, ma'am. Cancer relapse occurs in some patients. Do the therapies that you have discussed reduce or even nullify the chances of a relapse?
So that is a very good question. So the unfortunate thing is with all of these therapies, we haven't really reached a stage where we are able to see complete remission in our patients. So if you see complete remission in the patients and then you observe them for relapse, that is one thing. But with the chimeric antigen receptor therapies, there have been success cases where, like one of the success stories is a child actually who got CD19 CAR, and she was about 6 years when she got it. She's now 22 and she's cancer free. I can't say that it will prevent relapse because there could be some dormant cancer cells lying somewhere that we don't know of that can show up at some point. But I mean, the whole idea is to just control what is uncontrollable at the moment. So if we can get the majority of our patients to the stage where they have complete remission, then that is a big victory for us. Everything else will come after that. So sorry, I know that's not the best thing you want to hear, but.
Thank you, ma'am. The next question is from Ria, ma'am. Is there a way to decide which method of therapy would be accepted well by the patient?
Yes, so I don't think there is any particular way. So right now, our trials are open to all the two arms of treatment. One is the TIL therapy, that is just expanding tumor reactive T cells, and then the other is engineering the T cells to express specific TCRs. Our decisions right now, clinical decisions right now, are based mainly on time. How much time does the patient have? Because TIL is slightly faster because you're not engineering anything. You don't have to make a retroviral construct. You don't have to do all of that. But if a patient has some has the capability of withstanding some extra, like a month or two extra, then we do try to look for TCR therapy because it's more targeted. Our hope is that it is probably going to be more effective long term, but we haven't treated enough patients on that to make a clear determination as to one is better than the other. So it's still a work in progress.
Thank you, ma'am. The next question is from Dr. Jyoti. Will there be any psychological side effects of immunotherapies like depression? And how do you get the consent of the patients for these types of treatments?
So that is a very good question. There could be some psychological effects, more so because they get accompanying therapies, like, like the chemotherapy, the prior to administration of the T cells, or post they get IL-2 therapy. It is pretty daunting and heavy, and it can be depressing sometimes. And patients are looked into for all these triggers, and they are referred to the correct facilities for that. As far as consenting the patients, it is a open consent. So the entire clinical trial setup is read and clearly explained to the patient. You give them the pros and the cons, and then it is up to them and their families to make an educated decision. I will say it is not a very difficult sell, because the patients who come to us are pretty much at the end of all their options that are available outside, commercially available options. They haven't worked, and that's why they get referred to us, and that's why they come to us. So most of the time they will sign on pretty willingly, knowing that this may or may not work, but this is my best chance. So it is an informed, open consent. They know everything before they go in, and then they take the decision as to yes or no. And we do give them time. It's not like we pressurize them. They do get some time, but we do recommend that they decide sooner than later, because the faster we can get started in the lab, the better for them.
Thank you, ma'am. The next question is from Hazel D'Souza. Does immunotherapy increase your chances of survival?
So there isn't enough data to show that in general, but the hope is that it will, because the off-target effects are much lesser than just getting chemoradiation therapy, and it is your own immune cells. So that is the hope. That is what the trend looks like, but I can't say anything definitively, because we don't have enough data just yet.
Thank you, ma'am. The next question is again from Hazel D'Souza. What is actually better, immunotherapy or chemotherapy?
I don't know. Okay, so my personal bias would be immunotherapy, but I will not be able to say that one is better than the other definitively. Each one has its own advantages and disadvantages, which is why in combination, they probably work the best. So chemo alone may not be good enough. Immunotherapy alone may not be good enough. So chemo plus immunotherapy, that sounds more like the winning combination.
Thank you, ma'am. The next question is from Dr. Beatrice Sequeira. Will it be affordable for this treatment? How many settings do they need to go through?
So that is a great question. In terms of affordability, unfortunately, these treatments are going to be very expensive because just by raw materials that we need to make them in the lab is extremely expensive. But once they get fully commercialized, I'm sure there will be places we can cost cut and do a better job. But it is not an easy sell. Insurance companies here have a lot of problem with it. So, just because it's going to cost a lot more, and in general, cancer treatments are known to break the bank. So it is quite a expensive process. So the hope is that we over the course of research and years, we make it more affordable for everybody. As far as sittings, it's just one sitting. It's one time. So the patient comes in, gets resected. The resection is one sitting. Then the second time they get admitted is 15 days before the scheduled treatment to start chemotherapy. They take the immunotherapy. Within 3 days, they are sent back. And after that, it's all phone visit, like they do telemedicine. And if they have a problem, they come in. Sometimes for our studies, we ask them to come in for blood draws for us to study the persistence of our T cells in their circulation. But it is just one treatment session.
Thank you, ma'am. The next question is from Sanmeeta. Chemotherapy has a lot of side effects and reduces the immunity of the patient after repeated cycles of treatment. Are there any similar side effects seen in immunotherapy?
So far, no, because so the thing is, when we give the treatment, it's not going to last in your blood forever. But the idea is we give you enough that it is efficient enough to kickstart an effect that will sustain for a prolonged period of time. So that's the idea. So I don't believe those are any of the side effects that we can see with that we see in chemotherapy will hold good here.
Thank you, ma'am. We have one last question from Ria, ma'am. Have you witnessed any success stories with the immunotherapies in question?
Yes, we do have some patients who are doing very well right now that we are following. So we do have, and there have been cases outside our branch as well with some success. So yes.
Thank you, ma'am. So here we come to an end of today's session. I would like to take this opportunity to thank our eminent speaker for today, Dr. Nivedita Mohan Ratnam, for giving us such an enlightening talk. I would also like to express my sincere gratitude towards the entire team for working effortlessly. Last but not the least, I would also like to thank the audience for being present with us and making the session a huge success. Now, I request all the participants to please turn on their videos for a picture. I hope to see you all tomorrow, that is on 21st of January at 4 p.m. for an equally exciting and interesting session. Have a great evening ahead and stay safe. Thank you.
Thank you.
Devi, excellent.
Thank you so much, ma'am.
Yeah, all the speakers were good, and something that I'm so happy. I was just telling Ria that all my students, all our students